All right, so welcome to everybody to our last and final March Monday module session, which is on culinary macromolecules tonight. And we're excited about this. This is a new lesson, and it's written primarily for culinary teachers, and it was written with culinary teachers over 2 summers. But it also, we chose this topic because it pertains, biology teachers also talk about macromolecules in a different context. And so you hear it come up in both classes. And as a former science teacher working with culinary teachers, I was thinking, oh, well, this is a nice bridging unit between the two. So I think for science teachers who would like to introduce more culinary concepts that are really accessible to students, this is a good place to start. And for culinary teachers or FCS teachers who are trying to infuse their lessons with a little more science content, this will work for you as well. It's also brand new and barely field tested. A lot of the lessons that have been made in the unit have been used. You know, they were contributed by teachers, so they have some history. But the unit as a whole hasn't really been field tested, and it's big and it's deep and there's a lot going on in it. So if you have any feedback, we're really happy to do that. And because it's all in Google Forms and a Google site, it's really easy to edit. And if you have a lot to say, I can, give you commenting or editing privileges on different documents. So just let me know, because this is really a team effort right here. So any questions before we get started? All right, by now, hey, Adam, do you?
I was just going to say that this is also kind of a foundation for food science units. So if anybody's getting into food science, I know that's something that is kind of a growing topic in high schools. And both of my parents are food scientists, and they're like, we need more students with food science backgrounds because it's a growing field and isn't going to shrink anytime soon. So.
Thank you. Yeah, absolutely. So there's no PowerPoint for this session, so I'm just going to share my screen and I'll walk you through. The idea is that we'll go through together one of the macromolecules and then give you time to look on your own and then meet back in a group and talk about what you found and ways that you do it, ways that you might have to share, things that you like, things that you don't like, whatever you want to talk about, and then we'll wrap up. All right, so as I say, by now, I'm sure you all know where the workbook is and how to find it. We'll circle back to, well, the sign in. If you signed in before, you just need to put another hash mark, and then there will be an evaluation at the end. The easiest way to find this lesson is just to go to our home site. And here it's kind of highlighted because it's new. And there are 2 ways to access this. First, I'll show you the teacher roadmap, which has some of the behind the scenes things, but the website is really was built for direct student access. So the teacher shows the overview, sort of tells about the lesson components. Every lesson is built using the same 5E model from BSCS that will be familiar to a lot of science teachers. I'm not sure how familiar they are to the culinary or FCS or food science teachers. This is a really nice way. This is one of my favorite ways to orient kind of any lesson. And so that we had such disparate things that we were trying to shove into this lesson that this helps have everything in the same frame. So every one of the macromolecules starts out with the End Engage, which is a short little PowerPoint quiz thing, very low stakes. Then there's an Explore activity, which is something that students would do hands-on. Then there's an Explain, and that's sort of the direct instruction portion where there's a lot of information on the website and pictures and reading background information. And then the Elaborate is where you will find a lot of different labs. And these labs are mostly culinary labs. If you have science labs that you want to throw in here, I'd be really open to adding those as well. But it's meant to be kind of a mix and match thing where you can use whatever works for you and don't use what doesn't work for you. And then the evaluate comes in a couple of different forms. There are comprehension questions that go along with many of the labs and activities. And then in addition to that, every macromolecule has kind of a comprehension or an evaluation grid where students can choose, depending on what you want them to choose from a number of different ways of being evaluated. And I'll show you that. It's, as I mentioned, a Google site. There's a folder that you can get to. Here's this document. And then these are all the wonderful teachers that we worked with to build this over a couple of summers. And speakers and advisors, they're Adam's parents, Mike and Cheryl Leishak. So spoke to us, which was really helpful. And then the big ideas and the driving questions and the hyperlinks to every portion of every lesson is found right here. But first, let's go to the website itself. And I thought I'd just walk you through the fat section, just because. Here you see what it looks like, and let's just dive in and do the engage. So, what do you know about fats? All fats are unhealthy. True, false? So it's just to get your students thinking about things and having sort of priming their brains for what it is that we're going to be doing. And then there's, the answer. You can click anywhere because it just goes to the next slide. Our bodies don't need fat. Clearly that's false. We do need fat in all sorts of different ways. And so this talks about that. Fat-free foods are the healthiest choice because a lot of people think if they're in the store, they're going to look for the fat-free option, and that's not necessarily true. To make fat-free foods taste good, they're often high in sugar, refined carbohydrates, salt, and/or filler ingredients. So the example of peanut butter, natural peanut butter is actually high in fat, but better for you than the reduced fat peanut butter in most instances. Our brains are almost 60% fat. I thought that was interesting, so I threw it in there. We do need fat, and especially in our brains. And then gram for gram, fats provide more energy than sugar. And this is true, and this is part of the lesson that will become important because there's some caloric density labs that go along with the fat section. So just the idea that fat contributes 9 grams or 9 calories per gram, whereas proteins and carbs, of course, are 4 calories per gram. So you take the quiz, you go back to the website, and there you are. The hands-on activity for the fat section. Actually, I'll back up any questions about the quiz. You could add things if you wanted. They're just meant to be kind of priming discussion points. All right, and then the question or the activity is called, is there fat in that? And this might be familiar to biology teachers because this is kind of a common macromolecule lab. It's kind of hard to test for fat, or I should say the easiest way to test for fat that you see quite often in labs is this test, which is the brown paper bag test in all its highfalutin, what it's called. So you take something that you think you may or may not have a large fat portion to it, you rub it on a paper bag, and then see how see-through that piece of paper becomes. So there's just an explanation. The instructions are here. Students are asked to swab some water on a paper bag, 2 or 3 known fatty foods, so butter or oil or things like that, and then 2 or 3 unknown foods. And if you're in a culinary lab, it's pretty easy to find things around if you're a biology teacher. It gets a little tricky because food in the science classroom is always not welcome, but as long as they understand that this is not to be eaten in a science lab, then that's okay. Then you let this square sit for 3 minutes, and then you basically arrange them from high to low fat, and it's a conversation about what has fat, were you surprised in any of that, and then some comprehension questions. And did this activity help you figure out if any of your unknown foods contain fat? And then look at the nutritional label, so go back and see did how you align those papers match up to the nutritional labels on the foods themselves? All right. So then comes the explanation, the background information, and what are fats and where they are found, and how the base unit of fat is a fatty acid, which is 1 glycerol, which is this backbone, and then 3 fatty acids. And the 3 fatty acids can be different kinds of fat, which was surprisingly new to me. I just thought that monounsaturated fats were all the same, but you can have 1 glycerol with saturated fat, an unsaturated fat, and a monounsaturated fat, all in the same fatty acid. So students do a little label reading, and then they learn about what the difference is between saturated, mono, and polyunsaturated fat. And they're all made of carbon-hydrogen chains. It just has to do with how the carbons and the hydrogens are linked together. So a monounsaturated fat has 1, i.e. mono, 1 place where there's a double bond. Polyunsaturated has 2 or more places where there's a double bond in these very long chains that are storing energy through the carbon-hydrogen bonds. And then we talk about the different kinds of fat and what they look like and where you find them, and some examples of different kinds. And then, as I mentioned, that the one fatty acid can have different sorts of fatty acid chains, or the carbon-hydrogen chains. And so butter, for example, is mostly saturated fat with a small amount of polyunsaturated and 30% monounsaturated. So you can see how these are made up. And then what is trans fat, and why has that become so problematic recently, and how is that made? But fat is bad for you, right? And then it goes into a little bit about fake fats, which I just think are interesting anyway, because how they're chemically made with a sucrose backbone instead of a glycerol backbone, and they pack in the glycerides in different ways. So then a little thing on olestra and the digestion problems. So that's the direct instruction. Students can read that. You could bounce off of that for a lesson or a lecture, however you would want to do that in your classroom. And then we get to the labs that are associated with this portion of the website or the lessons. And there are 3. So one of them is a fat in ground beef, where students get different, like 95% lean, 80% lean, just different amounts of fat in their ground beef. They weigh it out. They get half a pound per session, per group, and cook it, and then actually weigh the fat that gets rendered off of it, and then see what the meat is like that comes out. And so then they weigh both of those, and then they calculate the percentage of fat in the meat that they were given and compare it to the label to see how accurate it is. Worksheets, and then some comprehension questions based on some nutritional labels. That almost every lab has some sort of nutritional label aspect to it, which I think even in science is good practice, because I think it's a really good skill that shouldn't just be in the culinary classroom or health classroom. And then, anyway, some questions about that. So that's that lab. Oh, now I've got so many things open. The second lab is a replacing lab, which is a fat substitution lab, which is pretty common, I think, in culinary classes, where students use they get a cake mix, and then some use pure oil, some use like an applesauce. They get a secret ingredient and replace much of the fat in there, and then they do a taste test and see if they can figure out what the secret ingredient is, and then rate it for on a sensory evaluation worksheet. And then there's the big reveal of like, oh, well, I was using apricot baby food, and a lot of people like that just as well, and it tastes as good, and it really reduces the fat content quite a bit. So again, nutritional facts and some questions about that. And then the last lab for the fat section is on caloric density. So this idea that some foods are calorically dense, especially if they're high in fat, just because you've got 9 calories per every gram of the food compared to proteins and carbohydrates. And so students take blueberries, popcorn, gummy bears, split pea soup, and figure out the caloric density of each of those foods. And first they predict which they think is going to be the healthiest, and then figure out if caloric density is a good measure of that. So then they have to go to a website and find the weight, and they do some math to figure this out, the calories and weight, and then figure out caloric density of these 4 items. And then analyze the information by thinking of it as a quadrant, like what could we eat that would be the highest in nutrients and maybe the lowest in calories, if you're interested in that, or what of those things is really lowest in nutrients and highest in calories, and what do those look like? And then some comprehension questions. All right, so then for the evaluate, I'll show you one of these. It's a student choice grid, and there's something similar for each of the other macromolecules, where students go back to the driving questions and then can answer some or all of them depending on what you ask them to do. And then they can build a model or make an ad campaign or a graphic organizer. There are just a number of ways that they can show what they know. Feel free to add your own. I snuck this one in. I've yet to receive one, but ask your teacher to fill out a short survey about this lesson, and that's a freebie for them. If you use the lesson and fill out our survey, then your students get a free square on this choice grid. So there you have the fat section. The other 2 sections follow a very similar framework. And so now I'm just going to give you, I think, like 8 minutes to look through the protein section. The protein section is the biggest one. I also want to be clear that there's a 4th macromolecule, which are nucleic acids, that biology teachers know about and science teachers teach about, but we did not include that in the culinary macromolecules because they're not a source of food for us. So just to be clear that we know that there's a 4th one out there, we didn't include it in this set of lessons. All right, so maybe we could do feedback in turn order, which you can pass if you don't have anything to say, but maybe if we could just do it, I'll do it as you're on my screen.
So what I discovered when I went through that I haven't really done before is talking about protein sources from plants. So I was really getting down the rabbit hole with the looking at the plant and protein labs and the plant protein labs, yeah. And having kids do a little exploration. And well, my first question was, for the protein discovery lab, I was curious what dishes you're giving to the kids to have them taste, because I didn't see that on the lab sheet that went with it. I didn't see like a teacher portion that said anything about what dishes should be presented, or if it was just up for whatever.
Excellent point. Yeah, I have a comment in my section of find out from the teacher who wrote this. I'll follow up with her. But yes.
For the protein discovery, yeah. The protein data collection, I thought I would definitely use that in class. And I imagined possibly setting some of that or all of the list of items around the classroom and having them rotate through to do their own kind of fact finding, or setting it up somehow so they physically have to look at a nutrition label and do some calculating. but I also, I could just do it on the computer and have them do it that way. I would probably, instead of having them search the price per unit kind of thing, just have those available for them, just because I don't know how I would have them look it up online. But I liked that one too. And then I was just looking at a recipe in the plant protein recipe lab. Even though I probably wouldn't be able to use that one in biology class, it was just still an interesting part that I was interested in. So yeah, these are definitely labs that I would use. And we previously kind of introduced the idea of getting protein from plants when we had done another a unit that was about an elephant's diet, and just comparing, elephant diet versus a lion's diet. And so how are elephants able to get all these amino acids and things that they need to build their muscles and all that just from plants themselves. And so I like this because they like to talk about themselves. So it's better. They all love talking about food.
Yeah. Thank you. And I'll work on the things that you just mentioned, because those are important. Amy, do you have anything?
I like the engaged one with the pencil protein and the pipe cleaner one, because it feels like that's a simple way of showing protein clothing instead of doing the Play-Doh thumbtack activity you typically do. But I can see how this activity could make that easier for them to comprehend too.
And at the bottom of that page, there's a link. This was actually started as a real kind of pure biology. It was published in the American Biology Teacher magazine. So if you go down to that link, there's you can get wait, do you do teach biology, or?
I teach medical science.
Medical science.
LTW.
So if you want to get into the domains, you've got your handle domain and the wrap domain, and you can make it a lot more scientific if you'd like with the use of that.
Okay, perfect.
I'm glad you like that. Yeah, I like that one.
Yeah, I am. Sorry. I could not open that first link, and then I just went to the next one, and somehow I landed in the cheese making here and explored that a little more. And I have goats and I make cheese of their milk. So I'm now thinking how I can put that in and actually show them in reality what happens if I have time
especially if you're a cheesemaker yourself. That's fabulous.
Yes. Yeah. We have a basic, when we do in biology, chemistry of life, just all the 3 biomolecules and just telling them they're all made of little units. And we have one little lab in there, which is a starch lab, just proving starch. So I don't know how much time for personal experience this is, but I love this background with cheese making and what the protein in there is. We do talk about lactose intolerance later. So getting to other ingredients in milk, I guess that would be interesting. I need to wrap it around, but I like what I read through there. I like it and how it's explained, and bringing that, everybody eats cheese or would like to eat cheese. So getting a little bit of also the history and all that in, I like that. It's fun. So I need to think about how maybe that bringing in.
All right. Thank you. Thank you. Dr. McKay.
Well, I'm kind of tagging onto those two. Really, the pencil and the pipe cleaner is one I can see myself using even tomorrow, as a matter of fact. I like that one very much so. And I suspect the cheese making is going to come up before the end of the year as well. That also appealed to me as a good way to convey the topics. So yeah, that was pretty much.
You see that I use it. I didn't read through the unit, but for my DNA and gene expression, I always my little pipe cleaner with the beads here, but and then all that. Yeah. There's your protein.
And so many culinary practices are really because you disrupt the shape of that protein. So just to go back to that principle is really helpful and a way of sort of imbuing the science into the culinary classes.
Yes, I do a lot with Play-Doh, but I like the pipe cleaner and the.
Just when you said it again, pipe cleaner, all of a sudden it pops in my head, I think.
Yeah, and the pipe cleaner activity is really showing how it has a function, like the shape determines the function of the protein.
I wish there was more time to do this in a regular biology class, because that is what life is. I mean, that's what we eat, and yeah, it makes us awesome.
Yeah. Sherry, you're next. Were you finished?
Yes.
Dr. McKay? Yeah. Okay.
Yeah.
All right.
Well, being a culinary teacher, I find this really fascinating because we kind of touch on things, you know, with proteins and all of that and fat and everything. And because we run a catering program out of our classroom as well, we tend to get really involved in the cooking. And so it's really nice to have some new ways to kind of go back and link it all together, because that's what I feel like that partially we've been missing, is that, you know, we get really into it and then we have to stop and do cooking. Cooking kind of gets in the way. And so, but I definitely will do the cheese one with my students, because I think that's just such a cool thing to do, and so accessible to everybody. So they'll be fascinated by that. So I'm very thankful for that.
We did it last summer and it turned out okay. And then we had a artisanal cheesemaker come in and she's like, you started with a gallon of milk you got at Walmart. Well, good luck. But yeah, I mean, it worked. It wasn't the best cheese, but
so, yeah. What would be your recommendation then for milk?
Artisanal goats that you raise on your own property and milk.
Don't have those.
Yeah. So that's just the problem, that you can't really buy non-fermented or non-pasteurized milk in the store, and that's not the best way to start. But I think it still works. I mean, the lab definitely works. You can eat the cheese. It's not as good as some of the fresh cheeses that you can go out and buy from a cheesemaker, but there's not a lot you can do about it in a culinary classroom. So,
which is cool to see how this liquid becomes that solid structure.
Yeah. All right, and then Adam?
Yeah, that's one of the things that I love about this discussion on macromolecules is that when you're looking at the protein structure, you know, the amino acids are amino acids for pretty much all proteins, and that, you know, you take them apart, put them back together in the ways that marketing within food products can really manipulate you into thinking, you know, various things about their product, when it's like, well, a plant protein and animal protein aren't going to be that substantially different. And so I think when we think about, you know, how are we building our bodies with the food that we eat, it's something that's really fun to think about. And I think this project does a good job of getting at that.
So, all right, well, as I mentioned, the protein section is the biggest. We have fixed the link if you want to go back and look at the plant and animal protein part. And I'll dig through that, find some recipes or the foods that she actually used in those and share that. But I'm going to let you go on to the carbohydrate unit. And because all of you were here for blood sugar balance, it will look very familiar because we're introducing that in this context. So I'll just give you like 5 minutes to look through the carbohydrate section, or maybe 6, and then we'll come back and we'll talk about that. But it doesn't have as much information as the protein. Let's move on to the carb section, and let's just go in the same order, and then maybe we can debrief everything after we talk about the carb section. So Dana, do you have anything to report back or share?
Yeah, when I was going through and looking at kind of how we classify the different carbs, it's different in this than the way we had been doing it for the like the glucose unit that we'd done in the type 2 diabetes unit. And so instead of saying monosaccharides, disaccharides, and polysaccharides, you're classifying it with starches and sugars and fibers. And so I'm wondering how, like, if I'm sure they can make the correlation, but should I just be saying sugar, starches, and fibers instead of trying to teach them mono, di, and polysaccharides? Or if it matters, I don't know.
That's a great, and then on top of that, some people use complex and simple carbohydrates. So there are so many different ways of talking about. We settled on starches, sugar, and fiber because that's what's on the labels, the food labels.
Yeah.
So,
well, and it's funny. Just today I had up on my whiteboard because I showed them a video. Today we did blood glucose levels. And so I showed them a TEDx or a TED-Ed talk that was about the brain and the fats that are in the brain, and then just why eating affects your brain, I guess, is really what it came down to. And so they were classifying them the way you did here with sugars and starches and carbohydrates, or sugars and starches and fiber. And so I stopped for a minute and I wrote up on the board, mono and di, and then polysaccharides. And then they also mentioned in this video glycemic index, which we haven't talked about at all. And so then I went simple and complex and high glycemic and low glycemic, and like tried to give them all the information. So I think they can sort it out. Like if I give them notes and they write it in their notes and keep track of it and understand how they all mean the same, or they're all grouping in the same way. But yeah, there's just a lot of vocabulary that when I teach it to my non-English speaking students, I really need to be selective and think about what's the most useful for them to know, really, I guess in life, instead of just for my biology class. So yeah. And then I went down the rabbit hole of looking at the Bristol stool sample, whatever.
Couldn't help myself. Yeah, so that's, I think for lifelong lessons, whatever's on a food label is more impactful, but the biology unit, having been written for science, ends up with a different way of, and I would say it's up to you. Like the one lesson in the biology diabetes unit where they do the cutout and they make the fiber, the long thing, like that has a ton of vocabulary in it that if you want to just get rid of all of that and talk about it in the context of carbohydrates, the different kinds of carbohydrates, that's fine. It's fine with me. Yeah. I think the one thing they need to know is that glucose, where it comes from, and that it is a monosaccharide, and that's the simplest sugar and why it's so important.
Yeah, I agree.
But that's a really good observation. I hadn't even put that together, that they're talked about in really different ways. So
yeah,
thank you. Amy.
I liked the yeast feast lab. I was looking into that because then I did that a long time ago when I taught biology. And so that made me remember that. I was like, I remember doing that. But then having it connect to like the nutrition label and looking at carbohydrates, I thought that was really cool. Because I wouldn't have thought to connect it by myself. So that was neat.
Great. And I think that's a much more simplified version of yeast feast than Dana's done in the past, for example, that some of you might be familiar with. So hopefully it's a little bit more accessible. Astrid.
Can I ask you the yeast thing? I've never done that, but I read about it, and it just shows the little balloons. One balloon is up and two are down, kind of when you do with sugar yeast, with one of them or both of them. How fast does that go? In a 50-minute lesson, could you show it? See, I'm online, I have to demonstrate it. Just, would it be that fast or?
Yeah, it
really
is. I mean, it's
I need to do that.
Especially if you're not there. There's a one where we use tubes that you invert, and you're actually trying to measure the amount of carbon dioxide that's produced. So it's more quantitative. But if you're okay with just a qualitative,
Just a
does it
demonstration,
produce
yeah.
or doesn't, then
Because that's something that sticks with them, right?
yeah, I
I
would
see that.
say blow up the balloons first a few times so they're nice and stretchy, because
Thank you.
yeah, so do that first and then use a small quantity. Like you don't want to have a ton of air that it has to, or volume of the flask that it needs to fill
Small
up
balloons.
before it starts filling up the balloon. But generally it's a pretty quick, if they're warm enough, if the yeast is nice and warm and percolating.
Goes.
Um,
Okay, cool. I need to
Yeah.
do one dry run. Okay, I have, yeah, for the classification of the sugars, Dana, when I talk about or we do in biology, we talk about it under biomolecules.
Mhm.
And somehow biomolecules are always, tell them the big ones, the polymers of smaller units, and that's how I say starches and fibers. So it's the poly units that kind of make the main structure of living things. Is that wrong to say? And then it breaks down into the glucose, which can be built up into other things again. You see that more under the whole cycle of how
Yes.
these smaller units get broken down when we eat and get built up into what we need to grow and or create energy. I don't know, makes sense.
It's too bad the biological units aren't on a food label, that there's that disparate.
Yeah.
But yeah, thank you. And I'm conscious of the time here. Dr. McKay, I don't want to cut anybody off. So.
Thank you. Kind of a little bit of a repeat. The yeast feast lab also appealed to me. We've been making so much bread lately that I definitely have yeast on hand. And as many times as I've tried to explain the yeast interactions, we still seem to have some confusion on that. So yes.
And it's great just to mention that yeast is lactose intolerant. So it's really cool to give them milk, and you look on the label and it says that there's sugar in there, but then nothing happens. And so that's a nice show of, well, it's the lactose is the 2 rings, and they don't have the enzyme to split to release the glucose.
So the yeast too appealed to me a lot. I really like that. And we're going to be doing another bread unit here before too long. And just a little quick reminder to, do the yeast part first and everything, because it's easy to just slip right into the cooking and kind of forget about that. So good reminder.
And another thing with yeast feast, in cooking, a lot of times you proof the yeast by adding some sugar. You can't do that in yeast feast, because then as soon as you put sugar in there, they're going to go, and you won't be able to see, for instance, that they can't utilize lactose. So just a heads up on that one. Adam.
I don't have too much more to add, other than I think I just always really like talking about the influence of fiber on glucose absorption, and we tried to represent that in blood sugar balance. So it is something that you can see while playing the game. But other than that, nothing more to add. Thank you all for coming. Joan?
I checked on the 2 protein models, and they open on my computer. So now I wonder if it's something that I have on my computer that allows those to open, because there are huge files, but they're really cool if you can see them. But we'll look into that as well. Thank you so much for coming. Really, really happy to see all of your faces, our stalwart group of Monday participants. So really appreciate you being here, and let us know if you have any questions or feedback or comments, or you do something at works that doesn't work. That's great. If you'll allow me 30 seconds to share this picture. This is a teacher sent it who did the cheese making with her class, and I just loved the look of it, of watching that happen. So I'll leave you with that. All right, well, if there's nothing more, and thank you for sharing that, always happy to hear how that works. Good luck to all of you, and let us know if you have any questions and along the way, and
Thank you.
Yes.
Very much.
All right,
Very good
thanks for coming.
resources.
Yes, thank you.
Very interesting.
Thank
Thank you
you.
very much.
And share away. Tell everybody you know if you want to.
Okay,
All right. Bye, everybody.
Okay.
bye-bye.
Thank you.